In this video, we're going to talk about this idea that DNA replication is something called semi-conservative. So we're going to talk about what this means first, and it's not a difficult concept. And then on the next slide, we're going to talk about uh, two scientists that developed a pretty interesting experiment to prove that this is how it's done. So we're going to just look at those two things. So there were a couple of different ideas out there about how you copy DNA. One was the conservative replication, and conservative stated that if we have this yellow original strand, we would just make another strand completely of new nucleotides. Right? So here's our old one, and the new one is made up of all these new blue ones. So that was the idea out there. The other idea was there was something called dispersive replication, where sections of it would be copied. So maybe the top part, and then there'd be like a new middle part, and maybe there'd be a new middle part down here, as you can kind of see in this diagram. So that was also an idea. But there was a lot of support for this one, but nobody could really prove it, or at least provide an experiment to lend support for this idea. So semi-conservative replication, as you can see in this picture, semi-conservative replication basically says is that I have this original strand, and what I'm going to do is I'm going to conserve half of it. So when I do this, and this is what hopefully you are leaning towards is the right one, our new copies of DNA will consist of one old strand and then one blue new strand. So that's how DNA works. So that's the idea. And this is the correct one. But how do we know? So let's get to the how do we know. So there were two scientists named Meselson and Stahl, and they wanted to show that DNA was semi-conservative. So what they did was, let me use different colors here. What they did was they grew bacteria in a solution that could only use heavy nitrogen to make their DNA. What are you talking about, Mr. Fleming? So that means that heavy nitrogen is actually N15. So they used N15. So when they were building their bases, right, nitrogenous bases made of nitrogen, all the nitrogenous bases were made out of N15. And that made them slightly heavier. Whoops. So here's my nucleotide, and they all have bases made of N15. So all of their DNA, right, all of their DNA looked like a double strand of heavy, in this case, red DNA. Then what they did with these bacteria, they made them exposed to a new form of nitrogen to make their new bases. So what they did was they used a light nitrogen, and this light nitrogen is in the form of N14. So all the nucleotides were blue. So if I'm going to make a new strands of DNA in that first generation, right? So this is called the parent generation. A lot of times we call it like F naught for 0. After that first generation, I'm going to have DNA molecules made of light DNA, right? One strand made of only light, because that's the only option to add new nucleotides on. And the other half of it will consist of this red. So what they did was they centrifuged the DNA. Centrifuge means you spin it at very high speeds. And what they found was if I had a test tube here's my test tube, that the heavy DNA in the F, whoa, I want to use a different color. The heavy DNA was all at the bottom of the test tube because it was heavier. It was down here. But in the second generation here, actually it's the first generation, not the parent generation, so the F1 generation. In this generation, when we spun the test tube, they found out that the DNA was somewhere in the middle. Like it was right here. So we had this like band right here. I made it purple because that's a combination of red and blue. Then, if we allow them to separate again, so this one is going to divide or replicate into two pieces of DNA, and so will this one. So in this next generation, I will definitely have, right? I will definitely have the original red for one of them. However, when I split the other half, it's going to be blue. So I have the blue one down here. And the same here, right? I'll have a blue, and I'll also have a red. And then, because the only option for my new nucleotides is the light nitrogen, the N14, my new nucleotides will be, we'll form a blue one here, a blue one here, this another blue strand here with a light nitrogen, and like this. So that means the test tube for this group would look something like this. Maybe you can help me. Like how many are going to be down at the bottom, this red category? The answer is 0, right? Because there is no more totally red strands of DNA. The new color, or the new the strand that we have now is in this middle. That's a combination, right, between the two of them. So half of your molecules would be made of this mixed group, and then the other half will be made of this lighter nitrogen, this blue group. So you'll have a band up here, and that would be for the F2 generation. And then if you did it again, right, another F3 generation, what you would notice over time is that you get more and more of these blue ones that are completely blue, and you would only be with the original group that are a mixed, right? So that was the Meselson Stahl experiment to allow them to give support to that idea that DNA replication is semi-conservative.